Dear students, now let us talk about the steps of translation. There are three basic steps in translation. The first step is initiation. The second step is elongation. And the third step is termination. Let me tell you that translation is completed in 5-to 3- direction. Now it's clear to you that all the three procedures we have studied, the DNA replication, transcription and translation, all of them are completed in 5-to 3- direction, or you can say 5' to 3' direction. Now the first step is initiation. In prokaryotes, we are talking about prokaryotes. So in prokaryotes, the polypeptide synthesis or the protein synthesis will begin with the formation of initiation complex. First of all, there will be formation of initiation complex. Now what is initiation complex? It is basically the combination of transfer RNA with ribosomes and then the attachment of messenger RNA. First of all, a transfer RNA which has chemically modified methionine. The other name of this methionine is N-formyl methionine. So the transfer RNA with chemically modified methionine or N-formyl methionine, it will bind to small ribosomal subunit. Now let me make it clear to you, the small and the large ribosomal subunit. The ribosome consists of two parts. There is a small part and a large part. This is smaller unit and this is the larger ribosomal subunit. Now there are three portions of larger subunit. There is an A site, a P site and an E site. A site is known as amino acyl site. The P site is the peptidyl site and the E site is the exit site. So when the protein synthesis begins, the messenger RNA becomes attached here. The transfer RNA, the first tRNA, this will be attached here along with the amino acid. The messenger RNA is in 5-to 3- direction. The ribosome will move this way. The whole ribosome unit will move in this way, this direction, and messenger RNA will be removed in this direction. Transfer RNA will also move step by step in all the three parts. So the initiation complex will be formed that when first a transfer RNA molecule with a chemically modified methionine molecule will bind to the smaller ribosomal subunit at the P site, as you can see. Nearby there are two sites, as I've told you, the A site, amino acyl site, and the E site. This dot is showing the amino acid, it's not the another P. So amino acyl site and the exit site. The transfer RNA will leave its amino acid here and it will exit through this point. The initiation complex is guided by initiation factor. The initiation factor will bind to AUG here. First of all, an initiation factor will bind to AUG here, and then AUG is basically our codon, the start codon on messenger RNA. So initiation factor will bind to messenger RNA, and according to that, when this will bind, the transfer RNA will bring the methionine and it will bind to the P site. Another transfer RNA will come according to the next codon, and then it will be attached here. This amino acid will be shifted to that, and this transfer RNA will be exited. Now let's see. Here the initiation factor also binds to messenger RNA on AUG site. Basically this step occurs first and then this step. First of all, initiation factor will see this codon on messenger RNA, it will bind to this codon, and accordingly a transfer RNA will come which will have the chemically modified methionine and it will be attached. Now the second step. The step 2 is elongation. After the initiation complex is formed, the large ribosomal subunit. Now the large ribosomal subunit will properly bind to transfer RNA with the help of some proteins which are known as elongation factors. Here the process was starting, so the initiation factors were there. Now here elongation factors will help in the binding. Now when the transfer RNA is attached here and the codon is read, then the whole ribosome, it will move a little bit further. When it will move a little bit further, the next codon will be exposed, and according to that codon another transfer RNA will come here at the A site with its amino acid. Now the two amino acids, they are present at side by side, okay, and there will be a formation of bond. Due to this bond, this amino acid will be shifted to this one and this transfer RNA will become empty. The two amino acids are adjacent to each other, they undergo a chemical reaction which is catalyzed by large ribosomal subunit. So the function of large subunit is that it catalyzes the chemical reaction between two amino acids. So as a result, the initial methionine will be shifted to the next tRNA. It will be attached instead of this transfer RNA to this transfer RNA by a peptide bond, and this will be the second amino acid in the chain. Now the third step number 3 is, in this way this whole series of polypeptides will be formed and they will be attached to one another. So the ribosomes, it now moves, more nucleotides will be exposed, and then they are guided by the elongation factors, and then the transfer RNA will come at A site. They will the amino acid which are present here, they will be attached to the newly coming transfer RNA, and the empty transfer RNAs will exit from the E site, and in this way the whole process is repeated until the desired chain is grown or the desired length of the chain is achieved. Now the step 3 is termination. After the proper elongation and the chain is now produced, the next step is termination. So chain terminating nonsense codons. We have studied about nonsense codons, that when they appear, the whole process of translation stops. For example, UAA. The nonsense codons, they do not bind to any transfer RNA, and when these codons appear, the termination factors are activated. The termination factors or the release factors, they are activated, and then they release the newly synthesized polypeptide chain. So what happens? The initiation complex formation, then the elongation step, and then the termination step. In the elongation step, the always the previously existing amino acids, they will be attached to newly arriving amino acids, and the previous transfer RNA will be exited and the new transfer RNA takes its place. Now let us discuss in the form of a diagram.